So here's the big question that sits at the heart of this story. How in the world can one tiny microscopic particle wage a war against our incredibly complex immune system for years, sometimes decades, and actually win? That's the mystery we're unpacking today.
All right, first things first. Let's meet the main character in this biological drama. To really get a grip on its strategy, you have to understand what it is and what makes it so unique. So, HIV is a special kind of virus called a retrovirus, and honestly, that one word explains pretty much its entire game plan. See, unlike a lot of viruses that just use a cell to make copies, a retrovirus plays a much deeper game. It literally rewrites the host's genetic code. It splices its own blueprint right into our DNA, which means it permanently, fundamentally changes that cell forever.
Let's break down its toolkit. On the outside, you've got this envelope studded with protein spikes. Think of these as a master key, designed to unlock our cells. Inside that, there's a protective shell called the capsid. And within that, the real goods. The virus's blueprint, its RNA, and a set of 3 absolutely essential enzymes. You can think of these guys as the heist crew. Reverse transcriptase, the codebreaker, integrase, the safecracker, and protease, the finisher. These are the tools it's going to use for the takeover. So, we've met the invader, we've seen its tools. Now, let's watch the main event, the cellular hijack itself. This is where it all goes down. And what's its target? Well, HIV doesn't just pick any old cell. No, it goes right for the top. Its primary target is the CD4 T lymphocyte. This is a brilliant and, frankly, devastatingly effective strategy. Why? Because the CD4 cell is like the 4-star general of our entire immune response. It's the command center. So by taking out the commander, HIV plans to dismantle our whole army from the top down. The break-in is a super precise 3-step process. First up is attachment. That protein key we mentioned locks perfectly onto the surface of the T cell. Step 2, fusion. The virus's outer membrane just melts into the cell's membrane, and just like that, its core is inside. And now for the first really clever move, reverse transcription. The virus's genetic code is in RNA, right? But our cells run on DNA. So that first enzyme, reverse transcriptase, gets to work, rewriting the viral RNA into DNA, basically translating it into a language our cell can't help but understand. And then comes the most sinister step of all, integration. That second enzyme, integrase, takes the new viral DNA and shoves it right into our own chromosome. At this point, the infection is permanent. The cell is now a sleeper agent. It's forced into replication, using its own machinery to churn out endless viral parts. Then comes assembly and release, where new viruses are built, and then they bud off from the cell, stealing a piece of its membrane as a disguise. And finally, there's maturation. The last enzyme, protease, makes a few final snips, and boom, you have a brand new, fully infectious virus ready to do it all over again. But here's the thing, our body does not just sit back and let this happen. This invasion triggers a massive, all-out counterattack from our immune system. This first battle, which happens in the first few weeks after infection, is called acute HIV infection, and it is an absolute war between the virus, which is multiplying like crazy, and the full force of our immune system trying to shut it down. Our body's defense squad has some key players. You've got dendritic cells, they're the first responders. B cells start pumping out antibodies to tag the virus for destruction. And then you have the CD8 T cells. These are the killer cells that hunt down and destroy any of our own cells that have been infected. This fight is so intense that the body produces a flood of antibodies, which eventually become detectable in the blood. That's a process called seroconversion. And you know what all that internal warfare feels like on the outside? The flu. It's a clever disguise. The body thinks it's fighting off a simple bug, while HIV is quietly setting up a permanent base of operations. Now, after that initial, really fierce battle, something strange happens. The war seems to go cold. The infection enters this long, quiet, and incredibly deceptive phase, a silent siege. This timeline really shows HIV's long game. You see that initial spike in the viral load? Well, the immune system manages to fight it back down to a stable level called a set point. And for the next 10 years or so, on average, the amount of virus in the blood stays pretty low and stable. A person feels fine. But look at that other line. While the virus seems to be quiet, the CD4 cell count, the measure of our immune strength, is on a slow, steady, relentless decline. It's a war of attrition.
fought in silence, and the virus is slowly but surely winning. So why can't our amazing immune system just wipe it out? Well, HIV has two secret weapons. The first is latency. It can just go dormant, hiding inside our own DNA, totally invisible to our immune patrols. The second is mutation. It's incredibly sloppy when it makes copies of itself, which sounds bad, but it's actually a superpower. It creates a whole swarm of slightly different versions, meaning our killer cells can't keep up. The results? A stalemate. Our immune system works 24/7 to contain the active virus, but it can never ever get rid of that hidden, shape-shifting enemy. But a stalemate like this can't last forever. After years and years of this relentless siege and the slow, steady death of those crucial CD4 cells, the body's defenses eventually begin to crumble. And it all comes down to a single number, less than 200. When your count of CD4 cells drops below 200 cells per cubic millimeter of blood, that's the tipping point. The immune system is now considered critically compromised. And that is the clinical definition of AIDS, acquired immunodeficiency syndrome. It's really important to understand this isn't a different virus. It's the final stage of the damage that's been caused by years of untreated HIV. You're diagnosed either by hitting that critical CD4 number, or if you start developing specific illnesses that a healthy immune system would have no problem fighting off. And these are called opportunistic infections. We're talking about things like Kaposi's sarcoma, which is a type of cancer, or really severe infections like tuberculosis. Essentially, with the immune system's command center completely destroyed, all these other invaders that were once harmless can now become life-threatening. Now, that whole story of decline, that's what happens when HIV is left to its own devices. But that is not where the story ends today. Not at all. Because this is where science fights back. And the first step in that fight is diagnosis. Modern tests are incredibly good. They can look for a few different clues: the antibodies our body makes to fight the virus, little pieces of the virus itself called antigens, or they can even detect the virus's genetic code, its RNA, directly in your blood. Once someone is diagnosed, the absolute game changer is something called antiretroviral therapy, or ART, and its goal is actually really simple and incredibly powerful. Two things. One, push the viral load down so low that it becomes undetectable. And two, by doing that, you allow the immune system to finally recover and rebuild its army of CD4 cells. And this brings us to what is, without a doubt, the most important takeaway of this whole story. While we still don't have a cure for HIV, it is absolutely no longer a death sentence. With consistent, lifelong treatment, the infection becomes a manageable chronic condition, and a person can live a completely normal, healthy, long life. So we'll leave you with this to think about. The story of HIV is just an incredible masterclass in evolution and adaptation. Knowing how it so cleverly hijacks our own biology, what does this amazing, decades-long battle really teach us about the profound and incredibly delicate balance that always exists between a virus and its host?